Hey, if you're a pharmacy student, you know this already. A drug can't work if it can't get absorbed by the body. But even before that, there's a critical first step. It has to dissolve. So today, we're going to break down the key theories that explain how a solid pill actually turns into a solution. This is a super important concept, so let's dive right in. So here's what we've got on tap. We'll kick things off with why dissolution is such a big deal. Then we're going to walk through the 4 major theories that explain how it all happens, and we'll wrap it all up with the key things you absolutely need to know for your exams. Let's get started. Okay, first things first, why are we so obsessed with dissolution? Well, it's pretty simple, really. For most drugs out there, if they don't dissolve, they just don't work, period. It's the essential first move in the whole game. So what is dissolution, really? At its heart, it's just the process of a solid thing dissolving in a liquid. You can think of it as a mass transfer. Molecules of the drug are literally leaving the solid surface of the pill or particle and jumping into the liquid around it. This is the moment the drug becomes available for your body to actually use. And this diagram here, it lays it all out perfectly. You see, a solid tablet first has to break apart. That's disintegration. Then those smaller particles have to dissolve, which gets the drug into a solution right where it needs to be absorbed. Only then can it pass into the bloodstream. Now, for a lot of drugs, especially the ones that don't dissolve easily, this step right here is the slowest part of the whole process. It's the bottleneck. We call that the rate-determining step. All right, let's get into our first theory, and honestly, it's the big one. It's called the diffusion layer model, but you'll also hear it called the film theory. This is really the foundation for pretty much everything we're going to talk about. The core idea here is actually pretty cool. The theory says that when a solid drug particle is sitting in a liquid, a super thin, totally unstirred layer of that liquid forms right around its surface. Think of it like a little film. They call this the stagnant layer, and the entire process of dissolution depends on how the drug molecules get across this tiny, tiny barrier. The theory breaks it all down into 2 pretty simple steps. First, the drug dissolves right at the surface, and it quickly fills up that stagnant layer until it's completely saturated. Then comes the second step, which is way slower. The drug molecules have to diffuse from that highly concentrated layer out into the main body of the liquid, which we call the bulk solution. And because that second step is the slow one, it's our rate-determining step. Naturally, in science, we need a way to measure and predict all this. It started with something called the Noyes-Whitney equation, which was then tweaked by Nernst and Brunner into the version you see here. This formula is basically the mathematical engine of the film theory. It lets us put numbers to it and predict just how fast a drug will dissolve. So let's quickly unpack that equation. The rate of dissolution, dC/dt, depends on a few really key things. D is just how fast a molecule can move. A is the surface area of the drug particle. This is a big one. The bigger the area, the faster it dissolves. That's why we sometimes grind drugs into superfine powders. Then you have CS minus CB. That's the concentration gradient, the main driving force. And finally, there's H, the thickness of that stagnant layer. The thicker that barrier is, the slower the dissolution. Makes sense, right? Now, that concentration gradient part is absolutely crucial. Let's say you're in a lab, just dissolving a drug in a beaker. We call that non-sync conditions. The drug concentration in the bulk solution, CB, starts to build up. This makes the concentration gradient smaller, and the whole process slows down. But think about what happens in the human body. As soon as a drug molecule dissolves, it gets absorbed into the bloodstream. This keeps that CB value near 0, which means the concentration gradient stays huge and constant. We call those sync conditions, and that's what we try to copy in our lab tests. And this graph shows it all. That curved line, that's non-sync. See how it starts off fast but then flattens out as the gradient shrinks? But the straight line, that's zero-order dissolution under sync conditions. Because the driving force, the gradient, is constant, the rate of dissolution is also constant. This is exactly why it is so important to get our lab tests right. We have to maintain sync conditions if we want to predict how a drug will actually behave in a person. But wait a minute, the film theory has a pretty big assumption. It pretends that the surface area of the particle doesn't change. But we all know that's not true. As a particle dissolves, it gets smaller. So what happens then? How do we account for a shrinking particle? Well, that question leads us right to our next model, the Hixson-Crowell cube root law. This was literally designed to solve that exact problem of changing particle size. So instead of looking at concentration, this law looks at how the particle's mass or weight changes over time. What the equation basically says is that the rate at which the particle shrinks is predictable. It's a really clever model that works great as long as the particle keeps its same general shape, like a little sphere, as it gets smaller and smaller. Now, of course, this model has its own set of rules. It works best for perfect little spheres that dissolve nice and evenly. It's not so great for weird, irregular shapes or for porous particles. So you see, it's a step up from the film theory, but it's still a simplified version of what's really happening. Okay, so we have a model for a static layer and a model for a shrinking particle. But what if the whole interface is much more chaotic and dynamic? That brings us to our third idea, Danquart's model, also known as the surface renewal theory. This model wants you to picture something totally different. Forget a calm, stagnant layer. Danquart's theory imagines a much more turbulent environment. It says that little packets of fresh liquid are constantly swooping in from the bulk solution, landing on the drug surface, grabbing some drug molecules, and then getting swept away and replaced by new, fresh packets. In this model, the speed of this constant surface renewal is what controls the whole process. Now, all the models we've looked at so far have one thing in common. They all assume that the slow step, the bottleneck, is diffusion. But what if it's not? That's the exact question that our last theory, the interfacial barrier model, tries to answer. This model suggests that before a drug molecule can even think about diffusing away, it has to overcome an energy barrier. It's the energy that it takes for that one single molecule to literally break free from its crystal structure and to enter the liquid. So from this point of view, the real bottleneck isn't the journey away from the surface, it's the act of getting off the surface to begin with. Okay. Deep breath. We've just walked through the 4 major dissolution theories. Now, let's pull it all together and really focus on what's most important for you to remember, both for the real world and, of course, for your exams. If you only remember one single sentence from this entire explainer, please make it this one. If you want to create a lab test that actually predicts what will happen in a real live patient, you must run that test under sink conditions. That means you have to make sure the concentration of the dissolved drug in your test liquid stays super low, just like it does in the body where it's constantly being whisked away by the bloodstream. So for your exams, here's the bottom line. First, remember dissolution is the bottleneck for drugs that don't dissolve well. Second, know the classic film theory and its Noyes-Whitney equation. Third, understand that Hixson-Crowell is the update for shrinking particles. Fourth, know that Danquart's offers that cool surface renewal idea. And finally, never ever forget the importance of sink conditions for getting good in vitro in vivo correlation, or IV-IVC. You see, these aren't just abstract ideas. They're the fundamental principles we use to design medicines that